So it doesn't matter what type of organism we're talking about, they all have a need for energy and perform metabolic processes. Moving right along, we're going to compare some different types of life forms. So on this slide, we can see different images of bacteria. And bacteria are probably considered by most biologists to be the most numerous organisms on the planet. And they are very necessary organisms. Some of them can be harmful, but many of them are necessary for life to exist on Earth. So here's some close-up views of some different types of bacteria. And you can see there's a lot of variety in their shapes and extracellular structures that they have. You can see some are coiled, some are spherical, some are rod-shaped, some are circular shaped. So they are highly diverse within their own class. Archaeans are very similar to bacteria in the sense that they look like them, but in all reality they're more closely related to what we call eukaryotes than bacteria. So these are two types of archaeans. The orange one is from volcanic ocean settlements, and the bluish one is from hydrothermal vents found on the floor of the sea. Once we leave bacteria and archaeans, we get into eukarya, which is the third domain of life. And here we can see some protists. And you can see protists are very diverse in their appearance. They are common to us in the sense that they're based upon the same cellular structure as we are, and that is eukarya. But even though they are similar, they are greatly diverse. Plants are another type of eukarya. They're a multicellular structures, and they are based upon the cellular structure known as a plant cell. Fungi are also eukaryotes, as are organisms of the animal kingdom. And even within the animal kingdom, we see a large array of diversity. So to summarize, there's a three-domain system that all life falls into: bacteria, archaea, and eukarya. And the eukarya domain includes the protists, the plants, the fungi, and the animals. Key differences. Bacteria, again, are typically organisms that are only based upon one cell. They do not possess a nucleus, and they are considered to be the most ancient of lineage, whereas the archaea are single cell, don't possess a nucleus, but evolutionarily they appear to be closer to eukaryotes than bacteria do. And then the eukarya are membrane-bound cells, so they have a lot of compartmentalization within them, a lot of different and diverse organelles that we'll talk about in week 2. And these can be either single-celled or multi-celled organisms. And again, they make up our protists, plants, fungi, and animals. When we look at the diversity of life, it can get really complicated. So scientists have devised a naming system. And the naming system, from very broad to specific, is organized as domain, kingdom, phylum, class, order, family, genus, and species. The genus and species together make up the name of the organism. So for example, all the way to the right, we see a Rosa canina, or a dog rose. Inductive and deductive reasoning are two forms of reasoning that scientists use. Inductive reasoning is a type of reasoning where you can arrive at a conclusion through an observation. So in essence, you're obtaining general principles from specific observations. So an example that I'm providing is based upon years of study of microscopy of organisms, so looking at the details, the small details, which organisms are built, a biologist concludes that all organisms are made up of cells because the scientist has looked at multiple organisms and seen these cellular structures. Deductive reasoning is where you're using a hypothesis to make a prediction. So you take a general premise, typically through observation, and you draw a specific conclusion. So for example, if all organisms are made up of cells and humans are organisms, then humans also must be made up of cells. Which leads us right into the scientific method. The scientific method is the way scientists test their hypotheses. So they make an observation, and from that observation, they come up with a hypothesis, which is typically like we just saw in the if-then statement. From that hypothesis, they design an experiment to test it, to see if the data that they collect will either support or refute that hypothesis. And once they collect that data and they process that data, they basically mathematically look at that data. They can come up with a conclusion as to whether that data supports or refutes their hypothesis. One such investigation in your textbook is the observation that olestra causes intestinal cramps. So scientists observed that when olestra came on the market, a lot of people started complaining about intestinal cramps. So they wanted to know if there was a link between olestra and intestinal cramps. So they designed an experiment, and in the experiment they created a control group and an experimental group. The control group is basically your comparison group. So that's the group where in this case, the individuals, the people, are not eating olestra. And then the experimental group is the one that eats the olestra-filled potato chips. So they're the ones that the independent variable, which is what the scientist is changing, is implemented, which is olestra. The scientist runs their investigation, in this case the people eating the potato chips, whether they are regular potato chips or olestra potato chips. And they gather their results. So in the results, 93 of the 529 people tested and in the experimental group, that's the group that undertook the independent variable of eating the potato chips with olestra, 89 of the 563 people had intestinal cramps later after consumption. So that's only 15.8%. So as we can see, the 15.8% is less than 17.6%. So a conclusion was arrived at that the percentages are not that different. You would think that if potato chips that were cooked in olestra actually caused intestinal cramps, that the experimental group of people who consumed the olestra-filled potato chips would have higher incidence of cramps, and they did not. So the results do not support the hypothesis that olestra causes the intestinal cramps. In this experimental design, we can see that we have observation of intestinal cramps and olestra coming onto the market. We devise a hypothesis. In essence, is olestra causing intestinal cramps? We design our investigation to have a control group and an experimental group. The experimental group is the one that is being tested for the olestra. So they're the ones that, in this case, eat the olestra-filled potato chips. As an investigator designs an experiment, they implement what's called their independent variable. The independent variable is what the scientist is changing, in this case, potato chips cooked in olestra and potato chips not cooked in olestra. The dependent variable is the data that the investigator collects as a result of the implementation of the independent variable, and that in this case is people stating whether they had intestinal cramps or not. So the dependent variable are intestinal cramps. And because the investigator has set up an experimental and a control group, this gives the investigator basically a baseline. The control group gives them a baseline so that they can compare their experimental results back to their control results. I hope that this has been helpful, and I look forward to speaking with you about week 2.